All right. Well, welcome everyone. Thank you so much for joining us. I am excited to be here to introduce today's topic. If you joined us earlier this month, we focused on the clinical side of this topic, and now we're here to focus on the research side. So this webinar is titled Research into the Genetics of Unknown Ataxia Without a Family History. And we have Dr. Fogel here to speak on that. So if you go to the next slide, Dr. Fogel, I will mention a few housekeeping things. So go back one more. Just a couple friendly reminders. You can turn on closed captioning using the CC icon from your menu on Zoom. Feel free to use the chat to chat with others during the webinar, but questions will be read from the question and answer portion of your bar. So please put questions there and we will get to those at the end. All right, next slide. So I'm very happy to introduce Dr. Fogel. He is a professor in the Department of Neurology and Human Genetics at UCLA, where he serves as the director of the Neurogenetics Clinic and the UCLA Clinical Neurogenomics Research Center. He specializes in the diagnosis and care of patients with genetic neurological disorders, including cerebellar ataxia and ataxia with oculomotor apraxia, spastic paraplegia, leukodystrophies, and many others. He runs an active research program to identify rare and novel genetic causes of neurological diseases and understand how best to use genomic information and technologies to efficiently diagnose and to improve care and treatment of patients. So welcome, Dr. Fogel. I'll let you take it from here.
Thank you so much for the really nice introduction. And thanks everybody for joining. This is my disclosure slide. I don't have any relevant disclosures for this talk. So I think I'm going to start off and just make sure everybody's on the same page. We had a really nice talk earlier this month from Dr. Wilmot, who sort of went through the clinical aspects of cerebellar ataxia. I believe during his talk, he said he wasn't going to be able to talk about the clinical stuff without talking about the genetics. And I'm also not going to be able to talk about the genetics without talking about the clinical. So hopefully we'll put it all together by the end. So the cerebellum sits here right above the brainstem, right behind the cerebral cortex, and what it does is it essentially mediates the interconnectivity between all of these different structures. Now, inputs come in, outputs come in, and basically what this does, it regulates timing of thought and action. It integrates all of our senses, vision, sensory, tactile sensation, proprioception or vibration sense, other environmental cues, and it basically regulates smoothness. And so what happens is if the cerebellum becomes damaged, then this input is lost. So all these signals are coming in, but it's not getting the information out that you really want. So what that leads to is it leads to an impairment of balance and coordination. And so the technical definition, the one that doctors use for ataxia, is an impairment of balance and coordination not related to muscle weakness. So if your muscles are weak, you're going to be have poor balance and be discoordinated, and it's hard to separate those two things out. But there's three ways you can get ataxia, and it's deficiency in one of three areas. One is the sensory pathways that regulate joint position sense. And then also the vestibular system, or the inner gyroscope within your ear, or the cerebellum. And really what we're going to be focusing on today is sort of talking about things that affect the cerebellum. But a lot of times, one or both of these pathways can also be involved as well. And the symptoms, everybody's familiar with these. They are the symptoms that reflect this coordination of voluntary movement. They're the eye movement abnormalities, trouble swallowing, trouble speaking, trouble with fine motor movement. And usually the first thing and one of the most prominent things is trouble walking, and everyone's familiar with this, so I won't go into that anymore. But the most important thing about this is to remember that ataxia is a symptom. It's not the disease itself. Ataxia is caused by something. So having ataxia tells you about a symptom, but it doesn't tell you about what caused it. It doesn't tell you about how bad it is or going to be, and it doesn't tell you about what the future holds. And the equivalent analogy to that, particularly in these times, is a cough. So everybody knows what a cough is. A cough is a symptom, but a cough can be caused by many, many, many things. You could have a respiratory infection like a common cold. You could have the flu. You could have a pneumonia, bacterial pneumonia. You could have tuberculosis. You could have a fungal infection, or, these days we always have to think about COVID. But these are all different, and their cause is different, their treatment is different, their severity is different, and the prognosis is different. The same is true for ataxia. So thinking about that, again, this is sort of my comprehensive view before we really drill down into the genetics, is there's a lot of things out there that cause ataxia. And there's things that people pick up over their lifetimes. There's things that people are born with, and there's things that we just really don't know the answer to. And probably the things in this group fall into one of these two categories. We just haven't figured it out yet. But just to get everybody on the same page with the terminology that I'm going to use, if we're talking about familial or hereditary ataxia, that's going to be ataxia that definitely has a known genetic cause, versus an acquired ataxia, which is going to be something that's not genetic. Now, there's a term that gets thrown out a lot, and I'm going to use it a lot, sporadic ataxia. And that basically means the ataxia came out of the blue. It's that we don't know why, we weren't expecting it, it just suddenly showed up. And when we're talking today about ataxia without family history, and that's really what sporadic means. It means there was no expectation of it, there was no family history. But the thing about it is it doesn't necessarily have to be any one particular cause. It can be any cause. So you can have a sporadic ataxia that's acquired, you can have a sporadic ataxia that's genetic. And what I'm going to do is I'm going to talk quite a bit about our work studying sporadic ataxia and identifying genetic causes that lead to that. I mean, for this audience, we don't need to talk about the numbers, but just to give a sort of a sense of this, about 1 in 5,000 people in the world has ataxia, and about half of those people have a genetic ataxia. So understanding the genetics is very, very important in being able to get to the root cause of ataxia for a lot of people. So again, why do we want to do this? I'm gonna harp on this point quite a bit, but the way that medicine has approached disease for a long, long time is this concept here, is that you basically have a bunch of people who have some kind of a shared condition. Let's say it's ataxia, and you define that and you give them a diagnosis and you say, oh, everybody has ataxia. You presume they all have it for the same cause. Once you have defined that, you give everybody the same treatment and everybody's going to get better, right? That doesn't work. We know this doesn't work, particularly for ataxia. And so what's a better approach? So one of the things is that we know that there's different reasons people get ataxia, and there's this term diagnostic reservoirs, and so ataxia is something that's sort of a diagnostic reservoir. People are given that diagnosis, and there is a more specific diagnosis they can get, like a specific genetic diagnosis, if somebody looks for it. And so the rationale for looking for it is, so you take the same people, but now you take what we call a precision health approach. And the idea is that everybody has their own unique specific diagnosis that we can identify. And from that, we can now do a number of different things that's tailored to a specific person. We can individualize their treatment. Maybe their condition has things that haven't happened to them yet, and we want to make sure that we know if they do or try to avoid them from happening, so we can look for those things. We can provide them and their family proper genetic counseling. There's a lot of benefits from knowing the cause and why something happened also. And lastly, and this is what we're aiming for, is that if we have a specific diagnosis, there is a very specific treatment we might be able to give that could potentially either stop the condition or hopefully maybe even cure it. And again, you know, I said I was going to hit this point with a hammer, and I definitely am. But this is an illustration that basically just shows the course of any disease. It could be anything in the world. And I just want to use this to illustrate why we want to know the specific genetic cause of someone's ataxia. So following this chart, you have some kind of cause. It can be anything, and here we're talking about genetics. So let's say it's a genetic cause. There's some period of time while, you know, things are cooking in the background, but there's really nothing that the disease hasn't really started yet. And this can be short, this can be long. It depends on what the underlying cause is. The disease starts, you get some period of time before symptoms begin, and then there's some period of time before symptoms begin, before you see a doctor for it. And then at some point you get a diagnosis. My personal favorite part of this whole diagram is how teeny tiny this window is for time to diagnosis. I'm sure everybody here can appreciate that is not drawn to scale. And the whole reason we want to do this is we want to avoid what's down here at the end. And this is disability or, you know, or a loss of years of life. And that's what we really want to avoid. So by, you know, so the things that we want to focus on here is this time to diagnosis. This can be decades for some people, and for really rare diseases, it might be never. So it's very important that we be able to recognize that, to minimize this time. If we know what the cause is, and we know the cause is genetic, that stops non-productive search for other causes. We don't need a million tests, you know, every doctor visit. We don't need a million MRIs. We don't need all of these extra things if we can understand what the underlying cause is. Then also, you know, as I said before, we can provide proper counseling and say, okay, here's what the cause is. Here's what that means for a person and their family. And most importantly, we hope to be able to modify this and arrest everything some point here so that it never gets to here. So this is really what the focus is for all of the research that we do in this space. So this is my chart. I use this for physicians. So I am not going to go through this. This is just simply for illustrative purposes, but this is a chart of basically how you evaluate a patient for acquired causes of ataxia. And so I expect like all of our physicians in training to know how to do this and things like that. But what I put it here to impress the point that this is not easy. It's not easy to read. It's not easy for physicians who don't think about this stuff every day to do. And so that's why seeing a specialist in ataxia is so important. You know, we've heard that said many times in many different contexts, but here's just one more. So the key points that I want you to take away from this are: there's lots of causes to ataxia, lots of tests that you can do. Not everybody needs every single test that's on this, but you need to see somebody who can understand what all these tests are for and why you need them, because the things on this list are a lot more treatable and a lot more modifiable than the stuff we're going to talk about next. And the earlier it's found, the better the outcome is. To really break this into simple things, there's really basic tests that everybody should get, which really cover these really common causes of ataxia. And then there's these much more advanced tests that really depend on an individual person's individual person's situation. Years ago, probably about maybe about a decade ago, we did a study where we surveyed physicians to how many patients got these basic tests before they came to see a specialist. And it was very low at the time. It was, I hate to say it, but it was actually none. None of the patients who were referred to our clinic actually had all the basic tests. These days, it is much, much better as physicians become more knowledgeable in what's the minimum everybody needs before going to see a specialist. So that's really, really reassuring that we're capturing a lot more of these things earlier. But again, it's it's just to be aware that this really needs to happen for everyone. And I'm not going to talk about this anymore. Dr. Wilmot did a fantastic job earlier this month, and I'm just going to leave the clinical stuff for now and go into the genetics and the research. So there's some certain things that we think about as physicians as to why someone might have a genetic disease. And the key thing is positive family history. Now, we're talking today about what do you do if you don't have a positive family history? And so that's really what we're going to focus on today. But that's really challenging for a lot of folks to think of genetic things because family history is usually the most telling clue. Obviously, if there's similarity to a known genetic syndrome, that's going to raise red flags to people. The problem is ataxia is very clinically heterogeneous, that all the ataxic disorders, for the most part, look pretty similar to one another. And it's very rare. It's very rare for someone to be able to look at a patient, no matter how good a clinician they are, and be able to say, this is the genetic condition that you have. Consanguinity, or people having their parents being closely related, that can certainly be a factor, but it's certainly not very common. Chronic progressive clinical course, again, that describes just about everybody. Or it's a rare disease commonly seen in a specific population. So you can see that these typical clues are not really going to apply to most ataxias. And then there's reasons why you might not find a family history, but there may be one to find, but you don't find it. So one is no one ever asks, because physicians are notoriously bad about asking people about their personal history. It makes people uncomfortable, and so sometimes people don't even bring it up. A lot of times people will ask the wrong question. They'll say, does anybody else have the exact same thing you have? And people will say, no, no one does. They're telling the truth. The issue is that the disease can look different in different people. And so if you don't ask the right question, there might be a family history to find. It's just that nobody looked closely enough at what conditions other people really did have. And everyone's guilty of that mistake. I've made that mistake myself. Information may not be available. People may be adopted. They may be not in contact with their family members. Other family members may not want to talk about their medical conditions. And there may be paternity issues. Every now and then we find that someone doesn't have a family history because they're not related to their parents the way that they thought that they were. The other thing is it could just be a feature of the genetics. I wrote here sideways inheritance. So when we think about genetic disease, a lot of times we think about it passing from parents to children to their children and so on. So that's one type of inheritance pattern. I'll talk more about that. But there's another type, which I'll bring up as well, where you don't see it in the parents, but you see it in multiple siblings. And that's due to recessive diseases. X-linked disorders can present like that. Mitochondrial disorders can present like that. So there can be unique features about the genetics that may make it not appear as if there isn't a family history, but yet there is. There can also be some genetic weirdness. There's certain things, I've listed some of them here. I don't need to go through them. Anticipation means that is usually, we talk about that with repeat expansion disorders, where depending on the size of the repeat, the disease might be milder or worse. Incomplete penetrance is a very annoying concept in that sometimes it's sort of the other way to think about that is sometimes the disease might skip a generation, or it might look very differently in one person and be much milder. There can be a number of other weird genetic reasons why it doesn't look necessarily like a genetic disease, and we always have to remember not to think, about genetics with blinders on, because there certainly are other things, and those other things are often more treatable. So now here, let's kind of dive right into it. So the autosomal dominant ataxias are what most people are most familiar about. This is what I was just talking about. So you have a parent who has the ataxia, and then about 50% of their kids will also have it. And this is because there's one affected gene, and there's a 50% chance of passing it down to each child. So about half of them will inherit it and ultimately get the same condition. And so these are the ones we know of as the spinocerebellar ataxias, or the SCAs. They roughly, it's about 4 people per 100,000 in the world are affected by these. They're, at the moment, using this SCA numbering system, there is roughly about 51 clinical types with 39 identified genes. The most common are the ones with the smallest numbers. So SCAs 1, 2, 3, 6, and 7, that represents about 50% of the dominance. Then everything else individually is less than 1%. And so this is just sort of a little timeline showing how quickly these are being discovered. And I'm going back to 2015 because one of the ones that we were involved in identifying, SCA 41, was back then. I'm going to actually talk about that a little bit later. But you can see how rapidly these are being identified all over the world. And in fact, just already this year, there is a brand new one, FGF 14. And there were 2 new ones in 2022, and these ones are all so new they don't even have numbers yet. Now then, there's these other ones that are recessive, and so this is sort of what I was talking about when I mentioned sort of the sideways inheritance. So in this case, each of the parents is a carrier for the condition, but neither of them actually has it. Now they each have a 50% chance of passing on that carrier status to a child. And so what ends up happening is you may get a child who ends up getting the carrier status from mom and from dad. And so because each of that reflects damage to the particular gene, that means 1 in 4 children statistically would be affected with the condition. These don't have the greatest naming and numbering system. They're also spinocerebellar ataxias, but they're recessive, so they're called SCAs. They're about as common as the dominants in the literature. They're probably actually more common because we keep finding more and more of these relative to the dominant ones. And again, depending on how you count, there's maybe 50 primary ones, but that number could go even higher depending on how you count, and not all of them have numbers. So the most common is Friedreich ataxia and RFC1, which was recently discovered back in 2019. And these 2 together represent at least 50%, or very close to 50%, I should say, of all the recessive ataxias. But these are also being rapidly identified, and this is just a list of the most recent ones going from 2015 to 2021. And there's a lot of other ones that have been identified in this time frame that aren't on this list because they aren't given a SCAR number. It's just referred to as the gene. And so I'm not listing all of those, but there's many other ones as well. Well, how many? So there are a lot of genes related to ataxia. So what this is, it's sort of an old slide, but it still illustrates the magnitude that we're talking about. So these are some really common neurological phenotypes. Here's ataxia here. And in terms of the number of genes that can cause this symptom, ataxia is second only to intellectual disability. So at the time this was made, it was just over 500. Now it's pretty close to 600 different genes have been identified that can cause ataxias, either a primary or secondary symptom. So it's a very large space to sort through. So that's why it's very challenging for clinicians and researchers to often pin down some of these causes. So I want to talk about some of the main techniques that we're using these days. And this is genomic sequencing, and you will have heard about this. And the two most common types that are done are exome and genome sequencing. So I just want to make sure that everybody is very clear on what these are and how they're done. So the genome is all the DNA that's in our body. It's all of our chromosomes and all 23 pairs of our chromosomes. And so you can think of the genome as a book that has 3.5 billion letters or 800 million words. Now, the way that genome sequencing works is, let's say you took that book and you took all the pages out and you gave it to 1,000 people, and everybody reads a couple of pages. And then everybody gets back together and they put the story back together. Now, if one person were to do it, it would take years. And so when the very first genome was sequenced, it took about 10 years to do it. Now this can be done easily within a day. And so this is sort of the method of how it's done. Now, exome sequencing is, let's say you have the same book, but you really want to get through it quickly. So all you do is you read the verbs. And so at that point, you're looking at just maybe 1% of the book. So now it's only about 70 million words. You still have the same 1,000 people who can read it. So now you can do it in maybe 1 to 2 hours. And so this is really the concept of exome sequencing. So in this case for DNA, the action is in the protein coding portion of the genome, which is about 1 to 2%. The rest of it is non-coding and acts as for structure and also to help regulate the expression of the protein in the other of the rest of it. And altogether, this represents 20,000 genes. So it's a very large search space, regardless of how you do it. So one of the things that got us into research in this field right from the beginning was this idea of, does exome sequencing actually improve ataxia diagnosis? So as we've talked about it, there's well over 500 genes that are associated with ataxia. Right now there's over 900 different phenotypes. They break down into all these different genetic forms. And then if you take a look here at this figure, which I like very much because it sort of illustrates the thinking about genetic disease from, before we had all these tools. And the idea was that you get this peak of genetic disease in childhood. These gray dots here. And then for adults, the thinking was that they weren't so much affected by it. And it was this black line here, which is multifactorial type things that wouldn't be so easily identified by genetic testing that was the cause. And the thinking was you wouldn't really be able to identify this very easy with genetic testing. So the thinking is, this is the familial type of stuff, and that's where all the genetic disease is. And then out here you have your adult patients. It's sporadic onset and very little value to genetic testing. So that's what we were really interested in. And in fact, what I'm going to tell you is that the line actually goes like this. There actually is quite a significant peak of genetic disease out here. This is a quick overview of what I just told you in terms of where the mutations are. And I'm not going to spend a whole lot of time on this because this is probably a little bit more technical than I want to get into. But aside from the most common genetic ataxias are all repeat expansions. And we'll talk about that a little bit more, but here's the dominant ones. They're all repeat expansions. Here's the most common recessive ones. They're all repeat expansions. But the bulk of that 600 other genes are all sequence changes that occur in the protein coding regions of these genes. So that's really the key point to take away from this. So our strategy for improving diagnosis is, can we make tests better? So this is a study that that we did very recently, basically trying to address that question. So this is a cohort of 184 subjects, and we don't need to go through all of the details about it, except to say they were evaluated for acquired causes and found to have none. They had negative testing for all of the common repeat expansion disorders, so we knew that they didn't have those. And the composition was primarily adult onset, so about 2/3 of them were adults, and 2/3 of them were sporadic onset. So that's just without a family history type of thing. So it's exactly the population that we want to look at here. And this is drawn from the Los Angeles area where we're located, and the demographics were pretty similar to what we see in our clinical population. This is our pipeline for genomic analysis. I put it up here not for anybody to have any understanding of this. It's too complicated even for me to show to physicians, but just to illustrate sort of the key aspects of it. And so we sequence all these folks. We do this very detailed analysis to try to find sequence changes that are going to lead to disease. But then we also can, using the same technology, we can also look for repeat expansion disorders using some new tools that have been developed within the last few years. And we can also look for these things called copy number variation, which is basically large deletions or duplications that, if they happen to involve an important gene, can also lead to disease. So we look for all of this together. And that's about as comprehensive as you can make this type of testing. And so here's basically what we found. So the key takeaway from this is, despite this population being primarily adult onset and primarily being sporadic onset with no family history, we were able to identify 47%, like nearly half of the folks with something relevant on their genetic sequencing. 24%, so about a quarter of them, were able to identify right off the bat and say, this is your diagnosis. Another 23%, we were able to say, okay, we found something that with some additional testing, maybe looking at some family members or maybe some other lab tests, we may be able to give you a specific diagnosis. So this is, we're very, very pleased with this number. The upside is that here's an additional 50% of this cohort that now has potential diagnosis. The downside is there's still 50% that don't. Now here's what's really key here. So this is a breakdown of the entire cohort by mode of inheritance. Either was there a family history or was there no family history? And then it's also, was it early onset or was it adult onset? Now, as from that graph that I showed you, the expectation was we would diagnose everybody who had a family history and an early onset. And we got about a third of people from both of those groups. So that's a really high number, just like was expected. And the expectation is you're not going to find a whole lot out here, but that's not the case. Case here is about 20% of people with no family history and about 20% of people with adult onset. We were able to identify a specific genetic diagnosis in those folks. So that's 1 in 5. Okay, it's not 1 in 3, but still 1 in 5. There's very few tests out there in the world that doctors can order where you can say, at baseline, we have a 1 in 5 chance of getting an answer if we do this test. So I'll take those off. Now, here's another key feature. Your DNA doesn't change, but our knowledge about how to read it does. So here's what happens if you go back and look 5 years later after you've sequenced a bunch of people. So this was an initial study we did on 76 folks, again, primarily adult onset and primarily sporadic onset. back in 2014. And we went back and we looked to see if now with the new knowledge we had, if we would diagnose more people. And so the key thing here is we actually reclassified about a quarter of the people differently than we had the first time. And the big takeaway is that 7% of these folks, almost getting to 10%, we were able to give a definitive diagnosis to after 5 years. So that is very, very significant. Now here's the key takeaway. This is a collection of different studies that use similar methodology from 2013 all the way up to our study at 2020. It's looking at diagnostic rate. It's looking at diagnostic rate in familial cases. And that's because those are going to have the highest likelihood of finding a genetic cause. And the reason I'm doing this is I want to illustrate that there's a diagnostic gap. Now, assuming every single person who's being studied here has a family history and they should have an identifiable genetic cause, we should get, given technical limitations, we should diagnose 96% of people. What do we really get? What we really get is we actually get 42%. And so that means that we are still missing half. So this is the line we want. This is where we are. This is the gap. And so even at our best, we can only get about 50%. So that means a few things. It means that, so let me hit the highlights first. So first of all, it means that this testing improves diagnosis for adults and early onset patients. Doesn't matter who you are, doesn't matter if you have a family history, this will improve diagnosis. We'll diagnose roughly 1 in 4 overall and give clinically relevant information to about half of the people. So this is something that really everybody should be considered for if you have an unknown ataxia at the end of the day. Reanalysis can improve diagnosis over time as our knowledge about changes in DNA improves. Adding things like copy number variation, repeat expansion can also improve the diagnostic rate. So basically meaning this test is getting better and better over time. But exome sequencing has a ceiling, it has an upper limit. And we think that upper limit is for about 50% of patients. And we don't know exactly why that is. There could be genes that we just simply don't know yet. And if that's the case, then we can maybe bump this up a little bit more. But the things that I worry about is there are mutation types that we don't know about, or other genetic changes that simply can't be seen by exome sequencing. And those are the things that we need to find in order to raise this number from 50% to 100%. So going back to this chart that I showed you before, so now in reality, we know that for this peak down here in childhood, where it's often familial, we can diagnose really up to half of folks under the right circumstances. But even out here, we can diagnose somewhere in the range of 20 to 40% of folks even without a family history. So we're getting there. Now, as I said, we need to also find new genes. So I'm going to very quickly give you an example of that. This was a gentleman we saw in our clinics, 40-year-old gentleman who'd been affected for 2 years. He'd been evaluated for everything, and there was just simply nothing that we could find. He had cerebellar atrophy on his MRI, and so we performed exome sequencing, and we identified this change in a gene that's called TRPC3. And I'll tell you what TRPC3 is in a minute. But first, I just want to tell you about why we focused in on that. Basically, it's highly conserved. So if you look here at this chart here, this is just all these letters here just represent different amino acids in the protein, and they represent a motif that is actually a functional domain in a number of types of proteins that are ion channels. They basically sit on the cell surface of cells and allow ions to flow back and forth. And so what you can see is, so these three up here are human genes. This is a fly gene, this is a fish gene, and this is a gene from worms. And so you can see this position right here is always an R. And in our patient, it was an H. And so we tested this by taking this gene, making the change in it, and putting it into the neurons of mice. And so what you see here is, these bars represent the number of cells that die. So this right here is normal gene after 24 hours and normal gene after 48 hours. And very, very few of the cells are dead, just a few percentages of them. And that just happens naturally when you grow them in culture. And here is the change that we found in the patient. So after 24 hours, about 10% of the cells were dead. And after 48 hours, between 50 and 60% of the cells were dead. So we thought that this clearly is something that is not doing what it should. And so this is what TRPC3 does. It's an ion channel that sits in the neurons that live in the cerebellum. And this is the pathway that it's involved in. It's regulation of the ion calcium. And so I'm not going to go through this whole pathway, but the reason that we were really honed in on this is because all of these things I've circled in red are known ataxia genes. So here you have SCA6, you have SCA14, you have SCA23, you have SCA15, 16, and 29 right here. This receptor here is actually targeted by antibodies in a form of acquired ataxia. So it was very reasonable to think that this would be a gene that was involved in disease. And so this is now actually SCA41, and this was the first case that we identified of it. But this is sort of the process about how that's done. And this work is a collaboration with Esther Becker's group at University of Oxford. The last thing I want to talk about is finding missing mutations. And so we talked a lot about repeat expansion disorders. Basically what these are is they're stretches of a particular nucleotide, often CAG, which encodes polyglutamine. So you may hear these called polyglutamine disorders or CAG disorders or polyQ disorders or all sorts of things. But you have this stretch of nucleotides, and normally they have a certain range where everything is just fine. But then if they grow to a certain size, then they start to cause a problem. And they can occur anywhere in a whole bunch of different genes. You can see them outlined here on this chart. And if they get too large, then you get some kind of disease. And a whole lot of common neurological conditions are caused by this type of mutation. Fragile X syndrome, people have heard about. Huntington disease, I'm sure people have heard about. And then an innumerable number of ataxias. And so in 2019, a new repeat expansion disorder was identified, and this was associated with a condition called CANVAS, which is a combination of ataxia, neuropathy, and vestibular dysfunction, which is the three systems that I told you right in the beginning relate to balance and coordination. Now, this was two different groups, one group in the UK and another group in New Zealand, and we were very fortunate to be able to work with those folks and be involved in this project. And the question we wanted to ask is, we wanted to know what the RFC prevalence was in North America, and so we actually did the first study to address that. And so this was collaborative work between my group and folks at University of Chicago and also at Brigham and Women's Hospital. And so we took a look at over 900 people. And again, the majority of them were adult onset and sporadic onset, so no family history. So this is, because again, this is the population we really, care the most about identifying. And so again, they were mostly screened, or all of them were screened for the most common repeat expansions. We called this a North American population because about 1/3 of the subjects from the University of Chicago were from Canada. So we've got the US and Canada represented here. The top phenotypes were basically your standard spinocerebellar ataxia, pure ataxia, or a number of folks had this phenotype called multiple system atrophy, which I won't go into, which is one of those idiopathic neurodegenerations that we don't fully understand yet. And this is just sort of a breakdown of the different cohorts, and they're all pretty similar. So the first thing we noticed there was a really high percentage of folks who were heterozygous, meaning they had one copy of an expansion RFC1, nearly 7%. In the original studies that were done, which were looking at quote unquote normal populations, the estimates were about 0.7 to 4%. So ours was much higher, but again, we're looking in a population of folks with ataxia. And so there's a number of reasons why we might have seen so many folks who were heterozygous. One could be that our assay was more sensitive than some of the other ones to choose. That's possible. The other thing could be is that there may be a risk of being a heterozygote in RFC1 to causing disease. And one of the things that that we know now from recent reports last year is that sequence changes on the other RFC1 allele, the one without the expansion, plus the expansion can actually also lead to disease. So we're in the process right now of going back through all of these folks and evaluating those possibilities. This is the conclusion. So going through this, so of course we had folks who had the canvas phenotype, and for the canvas phenotype, the majority of them tested positive for RFC1, but they actually weren't the most common patient in our group that actually did test positive, and in fact, the most common patient in our group were these folks here with ataxia and neuropathy. So that really represented about half of the people that we diagnosed positive. So the key takeaways were most of the folks were white ancestry, and that's really because of a founder effect from where this mutation comes from originally back way back in time. There was really no difference between folks with a family history or folks without a family history, so it was worth testing in either group. And then the key thing is, even though 73% of people with canvas tested positive, 22% of people with just ataxia and neuropathy also tested positive, and 1% of people with just straight out spinocerebellar ataxia tested positive. And overall, 3% of the entire cohort was positive. It doesn't seem like a big number, but this is actually quite a big number when most genes that cause ataxia are 1% or less. So at the end of the day, if you have ataxia and neuropathy, there is a 20% increase in the chance that you're RFC1 positive. And if you also have vestibular dysfunction, it goes up to about a 71% chance. And so our conclusion from this is that RFC1 should be considered in anybody who's undiagnosed, especially folks who have this canvas phenotype, neuropathy, or a chronic cough, which is something that's actually seen in this condition as well. And then it doesn't stop there. This is brand new, hot off the press from right at the end of 2022 and the end of 2023 is another brand new repeat expansion disorder was identified. This was in Australia, Canada, Germany, and India is where this has been found so far. Two groups were involved with this. Again, we were very fortunate to be able to work with one of these groups and to help identify this gene, and more to come. So the question is, so what happens next? So this is really the workflow. This is what I teach to all physicians about how this needs to be approached. And I'm going to go through this very, very quickly. We're getting short on time. I want to have people opportunities to ask questions, but the main thing is everybody needs a good clinical evaluation, preferably by an ataxia specialist if you can find one. They need to be thoroughly evaluated for non-genetic causes. Again, those are the most treatable. And then at that point, then people need to think very carefully about what to do. And if there's certain high yield testing, these are sort of like the most common ones. You test those first, and then you can get into some of the advanced testing. And what we've been talking about here is we've been talking about exome sequencing. Eventually, as the cost drops, this is going to go to genome sequencing, just simply because it's a more comprehensive test. We can do better at identifying some of these types of variants that we can't see as well with exome sequencing. And the key thing is, even if these tests don't turn up something positive, the data can be revisited at regular intervals. And over time, a percentage of those folks are going to be diagnosed. So how do we get there? So one thing we need to do is we need to share this information. And so one of the things that is very important is to have a repository where this genetic information can go so that people can use it. And the reasons we've talked about already is this ataxia is both clinically and genetically heterogeneous. 50% of patients that we think are genetic, we still don't know the cause. Mutations can be rare or private in families, meaning that you need a lot of people to find them. And we just really don't know all of the ways that these mutations can look. We don't know all of the ways they can affect people. And so the way to do that is to look at populations. And knowing precise diagnoses allows us to move forward with research, and allows us to get to the point where we can do clinical trials and things like gene therapies. So how do we go about doing this? So what we developed with support of the NAF is this thing called Centralized Ataxia Genomics Core. It's an ataxia-specific data repository with the purpose of being able to confirm and validate genetic variants and identify new genes that are important in ataxia. It's a resource that people around the country and around the world can utilize for identifying mutations in new genes. It's housed at the University of California, Los Angeles. We accept all types of sequencing, exome sequencing, genome sequencing. One thing that is unique about us is that we can accept HIPAA compliant data. So in some institutions consider exome or genome sequencing to be protected health information, and it can't go into public repositories. So our repository is HIPAA compliant. We can accept those sequences. And the Ataxia Global Initiative recognizes 3 data repositories for advancing clinical trial readiness and for gene discovery, and we are one of those 3 data repositories. So so projects like this are what's going to be necessary to advance this field even further. So just to summarize everything that I've talked about today, exome sequencing, genomic sequencing in general improves diagnosis for folks without family history. As our techniques get better, we're going to be able to diagnose more and more people. And as the cost of the testing come down, we will be able to apply it more broadly. Even if the testing is negative the first time, it may not always be negative. So we have to keep revisiting that. We're ultimately going to need to go into some of these newer technologies like genome sequencing and some of these other things that I didn't really get a chance to get into to be able to capture some of these mutations that we seem to be missing. And then also one thing I did not get into, which we're very involved in, is developing ways to validate these changes that we find on sequencing. And that's another key aspect that we just really didn't have time to dive into here. But all of these things are continuing, and hopefully will continue to reduce that number of missing diagnoses. And lastly, these are all the folks who contributed to all of the work that I showed. Too many to go through. I just want to highlight two people. This is Kathy No. She is my graduate student in my lab. She did all of the exome work that you saw. And this is Donna Siriani, who did all of the RFC work, or RFC1 work that I showed you. And thanking all of the folks who contributed and supported our research, including National Ataxia Foundation and all of the generous donors to our research. Thank you very much, and I'm happy to entertain any questions that anybody has.
Thank you so much, Dr. Fogel. That was very informative. There are lots of questions, and I realize we only have a couple minutes, so I tried to choose some that are as applicable to the topic as possible. I have one here. Do you see any crossover between genetic and acquired ataxias, perhaps an epigenetic expression? Particularly, I'm wondering if a genetic breakthrough might help those of us with an acquired ataxia.
Yeah, so epigenetics is woefully understudied in ataxia right now. Just for folks on the webinar who don't know what that is, basically it's a genetically heritable change that actually doesn't involve a sequence change. So like DNA methylation, for example. And certainly that does play a role. We just don't know how significant a role it does. I say it's under-recognized in ataxia. It's under-recognized probably in all disease for the most part, and people now are just starting to study that in more detail. So I'm certain things are going to come out of that research that are going to be useful to all folks.
Thank you. If my diagnosis was sporadic ataxia several years ago, would it be beneficial to have blood work done again to see if a now identifiable ataxia can be found?
So it's really hard to comment on an individual person's situation without kind of knowing all of the details about it. But I'll put it in sort of general terms. So sometimes people, even if it's known that they don't have a acquired cause, or if it's known they have a genetic cause, may still need testing for acquired things. Now, the reason being is because having a genetic cause doesn't make you immune to the acquired things. So sometimes people may suddenly get dramatically worse, and it may be because there is some other second problem that is influencing the first. So sometimes I'll see people who we are convinced they have something genetic, or we know they have something genetic, and I'll test a lot of the common acquired things again, because there could be a specific reason that they're getting worse that we can treat. So yeah, I wouldn't necessarily forget all of those things just because because they were tested once and were negative. But our knowledge of acquired ataxias really hasn't changed much in about 10 to 15 years. So for the most part, most of the things we look for now were the same things we were looking at then.
Thank you for answering those questions. We are at time, so we will go ahead and end there. Just a friendly reminder to all, what can you do next? You can become a member of NAF. Many of you likely already are. If you heard about this webinar, join a support group. It's a great way to learn about resources and make new friends. And of course, join the patient registry. So I don't know if you have any final words, Dr. Fogel, but I just wanted to thank you for your time and your very informative presentation.
Well, thank you very much for having me. And I know there's like lots and lots of questions, great questions we didn't get to, and I'll try to talk faster next time.
Thank you very much. Have a good one, everyone.
All right.